for getting me here. I'm not exactly sure who was responsible for that, but I'm very glad to be here. When I came, I didn't know anyone, and by now I've spoken to a lot of people, and it's been a very interesting introduction to the community of uncertainty quantification. So I'm a statistician working a lot with uncertainty, but I'm not technically from the uncertainty quantification community, because I think that's a bit of a parallel development that we are experiencing, where uncertainty is just occurring everywhere, so everybody has to find a way to deal with it. Originally, a long time ago I was in Germany and I did kind of pure math, and then I traveled around the world. I spent some time in the US as well, and then it's where I learned applied statistics. So the description was very long. You probably got that from my website or somewhere. I thought today, rather than picking one particular topic I'm working on that may or may not happen to interest people, I thought I'd give you an introduction in issues with sampling that occur in many projects. So mostly the examples will involve sampling from the real world, but the kind of biases that can come up, they can of course also occur when you simulate data, because biases, especially selection biases, they sneak in very easily without being aware of it. And in fact, that's triggered a bit the selection of this topic that just a couple of months ago I suddenly realized in one project we may have a big problem here with having overlooked something about the level where we actually sample, and maybe we're not getting the right results. So I have in the meantime sorted that out, and it will be one of the examples. So I hope there will be something for everybody, and I try to keep it non-technical, so there will be a lot of pictures and cartoon versions of the material. So it will. So the pointer didn't work, and now the errors don't work either. But we can do it that way. All right, so I'm going to talk about a couple of biases, and then the examples will just come up along with this. So this outline doesn't represent really the order. The first one is a bias called Bergson's bias. It's not so well known, and it's really weird. So I thought I'd introduce people to it. So what happens here is that you have a situation where you have two events, or it could be random variables as well, that are a priori independent. So A and B are not dependent of each other. So you may, but then if you condition it on both of them occur, on the event that at least one of them occurs, suddenly an dependency sneaks in, and the way it is phrased in the Bergson's paradox or Bergson's bias, it's actually a negative dependency. So why it's phrased like that has historical reasons. We'll get to the original example in a minute. So what happens is that you select something, and because you select something, you condition something, you actually introduce a dependency. And you can even have a stronger situation where you start out with a positive correlation, positive dependency, and then you end up reversing it. So that would be this case. That's a bit more awkward and less likely to happen, but even that could happen in some strange examples. So it's important to be aware of that. And then the other, I think I can still can I use it as a pointer? Actually, maybe. Oops. Oh, pointer, yeah. Oh, okay. Yeah, oh, brilliant. Thank you, sorry. I never get along with pointers. I don't know. It's some kind of allergy. So here is the same thing just for random variables. These are, I mean, this is in the event description. So event is just a yes or no situation. So that corresponds more to discrete data. And this would be the same kind of phenomenon just formulated for continuous data. So that can also work. Now I give you something a little more explicit about it, and that's the original example where the Bergson bias comes from. And it was observed in the clinical context, so in epidemiological studies, where they found that there was no correlation between a risk factor and a disease outside a hospital, but suddenly among the people who were admitted to the hospital for that disease, it turned out that there was a negative correlation between the risk factor and the disease. So that was not what they wanted to have, but so it was observed, and so so you can ask how. So in the historical example, it was diabetes, and the risk factor was diabetes, and the disease was called cholecystitis, whatever that is. They observed then that within the hospital, the risk factor had a negative correlation. People without that risk factor were less likely to have the disease. So the reason for that is, the reason that's being offered is that among those people who were hospitalized, they were hospitalized for some other factor, and that other factor would have probably caused that disease. So the fact that they had diabetes made it less likely they had some other factor, to put it that way. Now, a mathematical version of the story. So here's just the bias again in this technical way of probability language to write it up. So you have here a conditional probability just on B, you have independence, but if you also make that extra condition that A or B need to happen, then suddenly there's this negative dependency. So how can this happen? Well, in general, we can't just condition on stuff and assume that independence survives that conditioning. It's just a general fact in probability. You can't introduce conditions. It's very easy to generate examples where that goes wrong. So we couldn't have expected this to be stable. However, usually when those biases occur, you're still surprised that this went wrong. Now, how can we explain this within the formulas? Well, so if I condition here, this just means that because I already conditioned on B and then on the union, so it just means I'm conditioning on B. So if that is the same as A by assumption, we assumed independence, then if I'm thinking of that situation that the probability of A is actually smaller than the probability of A when this union happens. So this is the kind of situation that you need for this fallacy or bias, people call it paradox, all sorts of things. For that to happen, you need a situation like this. So this is basically mathematically what is happening. This is the reason why the conditioning destroys the initial independence. And now I'm giving you some other examples where that can occur. So first, here's just a kind of technical example. So we have a scatter plot. So this is now a continuous situation. You have two independent or nearly independent random variables, a slight positive correlation in this example. And then what you do is you just select stuff. So this is a sampling situation where you select things systematically based on, in this case, it's basically a condition on the sum. So you're kicking out everybody where the sum of these two random variables is too small. And now if you look at the rest of this, what's left has a small negative correlation. So you actually managed by erasing your data points to create this situation. Now, this could also happen if you actually sample from a population and you are somehow, for some reason, not taking care to cover the whole area. These are things that become more and more important with big data as well, because it's quite challenging to cover the whole space when things get more and more kind of difficult to overlook. Now, a more extreme version of the same, it seems all very artificial, but we'll see in a minute, we'll see an applied example where that actually happens. So you can make that even more uncomfortable, because if you have another condition, if you select these guys here, then if you take them out as well, you end up with a strong negative correlation. So, and now let's look at an example where this happens. There was some work it's a while ago, I think in the 70s, done by a person called Robin Dawes, who looked at graduate admissions in the US. That seems to have been a fun topic in the US in the 70s, because there's also this famous Berkeley study with graduate admissions, and the Simpson's paradox, but we're not talking about that today. This is much simpler. They just found that these two criteria they used for graduate admissions, two scores, they were normally positively correlated, but after they admitted students in the pool that was admitted, they suddenly became negatively correlated. It's actually quite intuitive. It makes sense because you're having such a cutoff here. But if you don't think about it, you can easily get things wrong by overlooking this, because you take it for granted that your correlation, your positive correlation, will stay. So this brings us to my favorite topic. Of course, that wasn't so serious. Undergraduate admissions in the UK. Quick introduction for those who are not in the UK, not from the UK, because it takes a bit of getting used to this. So in Europe, people just do their degree and do nothing until they have their grades, and then they try to find a university that wants them. This is a bit different here in this country. There's more planning going on. People start to apply to universities a year before or even longer, look at places, and then they gather conditional offers, and so they're based on predicted grades they get from their teachers, and then they get these conditional offers, and if they make the conditional offer, then they are in. However, there's kind of a random marketplace in August where people who didn't get their offers, they may still get in if they have some other thing to show. So that's a bit chaotic. It's actually, again, different in Oxbridge, where they have interviews. It gets too complex because they, again, do it differently from each other. But typically, most universities are what I initially said. So there is a lot of work for the universities all over the year to look at all these predicted grades and try to make sense of them, and decide who you give an offer and so on. And so we have also done lately a bit of pilot analysis on the relationship between the achieved grades at school and the outcomes at university. And this is what we really want. Yeah, we want to make the right decisions. So we're doing more of this analysis, but I came across the Bergson paradox because we actually found something like that going on. We suddenly had in one part of the data set, we found that a further mass B, people seem to be doing better than with a further mass A. So that shouldn't happen. And then we could explain it with this paradox and with a certain thing that happened that year and so on. But that showed me that this is not just some theoretical stuff that happens in probability spaces. It can happen to you in real life. So I've experienced that kind of paradox just recently when we looked at this data analysis, and I thought that was fun. So in particularly, if you're not Oxbridge, people may actually go elsewhere if they are at the very top. So you do have a situation where you can have this awkward reversal from a positive to a negative correlation, because you reject all these people. The very top ones who are good at both, they go to Oxbridge. So you have this funny negative correlation in the pool you're left with. Then I just thought I should mention Simpson's paradox. I don't know how many people actually know Simpson's paradox. Okay, so less than I thought. So you have to know it, because I love Simpson's paradox, and you can just explain it in a cartoon version. So here you have positive correlation, yeah, not very strong, but some positive correlation in a scatter plot, really bread and butter stuff. But then it turns out, if you have a structure in your data set where you have some other factor, in this case you have actually 3 different groups, it can happen that you have negative correlation within each group. It's a lot of fun to generate examples like that, and it's always a bit shocking. So here they have a strange example, because it's from a psychology paper. They tend to make a lot with these biases, so I got it from a psychology paper. So they have neuroticism, which really is a psychological criteria for personality. It's not some kind of weird word. It really is a technical work in psychology, so they have metrics to test for that. And then they have these three groups, low, medium, and high education, and correlate that with the salary. So for each individual group, there's a negative correlation. But if you pool all of them together, you suddenly get a positive correlation. This is why epidemiologists always are very careful with merging data, pooling data. So if you pool data, all sorts of things can happen, which is, again, related to our initial formula we had, where it was the other way around, where I said if I have independence, I can't just start conditioning on some stuff and hope the independence will survive that. That may not happen. Now, the Simpson's paradox is also called ecological regression, because that's where it came usually from different geographical regions corresponding to these subgroups. That's where the name comes from. And the more core Simpson's paradox is actually in discrete tables, just in 2x2 tables. And here, there are a lot of very complex examples on the web, and you need a lot of arithmetic to follow them. Here's a very simple one, and it's real data, that's why I like it. You can make them up, of course, but here's a very simple one. So it happens that in this data set from the 70s in Canada, they wanted to know who has more children, the French or the English-speaking people in Canada. So they compared that in the different provinces, and it turned out in Quebec, the French had more children, 1.8 per woman. It's the way they count that, I think, in demography. And the English had 1.64, so the French had more. In other provinces, also the French had more. But then, mysteriously, if you pull it, the English had more. So you can sit down, it's primary school mathematics, and you really don't want to believe it. How can that be true? You can sit down, do some calculation, which has to do with the size of the provinces. That is the arithmetic reason behind this, but it's still unintuitive. You want to not accept it, but you do the math, and it's possible. So that's Simpson's paradox. It's a paradox because it's unintuitive. In every province, the French have more babies than the English, but if you pool it all together in the whole of Canada, the rate is higher for the English. So you can argue that this is weird. But, I mean, we can explain it. So in that sense, after you explain it, it's not a paradox anymore. But it is still called Simpson's paradox in the community. This is how, I don't know another name for it, I think. But it's often with paradoxes that once you explain them, they're not really paradoxes anymore, but we still have the name paradox because we initially thought that was paradoxical. So let's now jump to some science. I want to talk a bit about microscopy pictures because they also have some related issue with correlation. And I work with a guy called Steve Royal in Warwick in the medical school. And he's very open to statistics, and he does also these beautiful pictures. So what they do is they have 2 channels or more to measure protein concentration in cells. And then what they really want to know is something about the interaction of the proteins. So what they try to do is they color them with fluorescent dye, and then they are developing scores that measure how strongly these interact. This is, however, not so trivial, because there is noise, there is empty space, and it's not, so all sorts of issues come in to do with sampling, to do with noise, to do with various things. So the typical way of measuring colocalization is the Pearson correlation, and it has the advantage of having a convenient mathematical property, scaling invariant, and so on. But a problem with this is they're actually ignoring the spatial information. So that is at the moment not yet quite tackled how to do it in a more spatial way. They just correlate the intensities and then they calculate the correlation. There are some alternative ways to do it, but they like Mander's coefficient, but that was invented by biologists and is very inconvenient mathematically. So for example, it's not scaling invariant. There's still work by statisticians done on this. Here's just some real-world stuff. So if you are lucky, you get a scatter plot like this one. It's very easy. It's nicely correlated and so on. But you could have something like this, and then you really don't know what's going on. It could be that if you get the noise out, maybe it's actually negatively correlated, but maybe something else is hiding here. Maybe it's a matter of subgroups that after you get the noise out, that is covering things, but that could be filtered out in a way. Then maybe you discover various subgroups where it is positive correlated. So that is a thing that needs to be analyzed more clearly. And here is an example where this kind of scenario is true. So here they have actually different layers of because the cells are three-dimensional. So when we have these microscopic pictures, we are actually simplifying it to slices. So if you have now this situation, they have done 3 different slices, different heights, and then they found linear correlation. When you color them separately, you find much better correlation for those subslides than if you pool it all together. So that is not Simpson's paradox in the sense, but it is the same issue with merging subgroups in scatter plots and changing the story, not in the sense that it reverses the correlation, but in the sense that it makes the correlation much stronger. Yeah, so let's have a quick look at sampling biases. So here's just a list, and there are a lot more sampling biases. Because the next thing I want to talk about is something called size bias or length bias sampling, and some examples for that. So you may have seen some of these. There's even one in astronomy, for example, where it turns out that bright objects are preferred. Yeah. So in then there's censoring. There's a caveman bias, which has to do with what is preserved and what is not preserved, because what we see of those times is what has been preserved in the caves. We don't know what kind of drawings they did on the trees because we can't see them anymore. So that's a very strong selection bias that people in archaeology have a lot of issues with selection bias. So let's look at size bias. So here's a cartoon version of size bias. Just think of catching fish. You do it with a net, and you're introducing a selection bias because you're going to miss the little ones. They fall through the net. So what you catch is giving you a different mean and a different distribution than what's really in the sea. So it's something to be aware of. It's just a nice visualization of sampling bias. Now here is length bias. Who is familiar with the waiting time paradox or the bus paradox? Which again is not a bus. It's not a paradox once you understand why it happens. So are you familiar with the problem that you stand on a bus stop and the bus doesn't come, and then you always have the feeling you wait there for too long, and something seems to be unfair? Yeah, no problem. We can explain this. It's true. You're absolutely right. Unless you're in Japan, where everything comes exactly on schedule. But as soon as you're in a country where some variation, your subjective feeling of being upset is actually correct, because what happens is that you're getting somehow not the deal you think. So you can model that mathematically. So it's important to have this in mind. It's not like you are selecting a bus and then you are calculating the time some other people would have had to wait if they had shown up here. That's a different thing. It's a different question. If you're in the bus center and you can randomly sample buses, that's nice, but that's not your position. Your position is you randomly occur at a bus stop, and you are here, and then buses come. Now, what you're doing is you are sampling. Basically, you randomly select these intervals, but you don't have the whole pool of buses. And so what happens here is, first, just intuitively, you're more likely to hit one of these long intervals. That's the problem. That's what happens to you. So, If they were all equally the same size, it wouldn't be a problem. But as soon as you have variation in those lengths, unfortunately, because of the length bias sampling with the fish, yeah, so this is a big fish here, so you're more likely to catch the big fish, long waiting time. So here is the technical background for that. So you can model this as a jump, so as a process. You know, you have these jump times, you count the number of buses, you have these inter-arrival times or holding times, S. And what you're interested in is this interval. How long do I have to wait? So you figure out, so this is actually called renewal process in microprocess theory. So you figure out where you are in which of these intervals, and you can determine where you are between these two jumps. And then you have your residual time, and you want to calculate the mean of this residual time or distribution of that. We call it y. And this is this distance that you have to wait. And then it turns out, I'm not doing the whole calculation here, but it's actually just a few lines. You can even find it on Wikipedia. It's just cumulative distribution functions. You can calculate that the expected value, so that's the average time you have to wait, is mu square plus sigma square divided by 2 mu, and mu and sigma are just the parameters of the cumulative distribution of these holding times, these inter-arrival times, these ones. So you would have thought that your deal of waiting is just the mean of this distribution, but unfortunately that's not the case, because this sigma square here, this variance, the variance just sneaks in there. So as soon as that variance is larger than 0, you have this bad deal of having to wait longer than you think. So that is the waiting time paradox, which is not a paradox anymore now. Yeah, so this is the thing summarized. So now let's actually apply this to something more serious than waiting for the bus, and this is cancer screening. So in cancer screening, you have a lot of different events happening. You have a cancer that is clinically detectable at some point, but before that, and that is why you do the screening program, you can detect it with X-ray screens. Yeah, those programs exist for various cancers, and they're talking about introducing them for some more cancers. So now you end up with different times. So you have a time where it is screen detectable, the cancer, and that's a bit earlier than the time where it's clinically detectable. And then you also have the times where these things are actually detected in a particular person. That's then again a different thing, because you're not going to a screening every day. Screenings happen every 1, 2, or 3 years, depending where you are. And there's a lot of debate about how often screening should happen. And this is part of the rationale for those kinds of for this kind of work, that you want to inform screening programs how often screening should happen. And so what can happen? So you have this time between the screen detectable and the clinically detectable. That's called sojourn time. And then you have the time that when you do, I'll just use this arrow, it's easier. When you have it, so there's a technical difference between screening and screen detectable. Forget about this. So you have a lead time, which is the time between the screen detection and the clinical detection, or when it would be clinically detectable. Yeah? So lead time means this is the time you gain because you have a screening program. So this sounds good, and for the most part it is also good, but how often to do the screening is actually a trade-off. And that has to do with a length bias. Because if you look at different cancers, you have cancers that grow very rapidly, and you have cancers that grow slowly. And now it turns out, because of this length bias, that the screening program will catch a lot more of the slow-growing cancers. But the slow-growing cancers are not the dangerous ones. In fact, some of them don't even need treatment. Some of them, if the person is already older, it grows very slowly. And some of them, it really depends on the type of cancer, but in breast cancer there's some some version that is quite harmless and people just wait and do nothing if they're already older, because when people are already older, doing treatment introduces a lot of risks for their life. Yeah? Chemotherapy can be extremely dangerous to people. When they're already fragile, you better do nothing. So because of that, we actually have a problem here with length bias sneaking into the screening program, and now it has various consequences. It has the consequence that that people get scared, even though they should have just waited and doing nothing if it was a slow growing cancer and no indication for doing anything. It also has the problem that it, for the statistics, that it gives us the illusion that we are saving, that we are gaining a lot of survival, and we actually don't. So just because the lead time now counts as survival, extra survival time, yeah. So in our records, we look better, even though the people die at the same time point. Yeah, so you can have people who die at the same time point, but on paper it looks better because they were earlier diagnosed, so they survived longer. Yeah, so there is an ongoing discussion, and different countries have different answers to that. Yeah, so we did with one student some work that used a lot of data from the UK that is trying to estimate some of these parameters, so germ time, lead time, sensitivity, and so on. So it gets a bit technical, but so you know this is actually ongoing research. Now I want to tell you one other application of that, which has to do with genomic testing. Who has heard about this company 23andMe? Okay, some people. Yeah, we can chat about it later, but I may spoil it because I'm probably a bit too much opposed to it. So you get a one-sided view now, because I've worked with genomic data, and so I don't trust it all so easily. But more importantly, let me give you a little bit of background about biology before I get to the connection to the length bias. So just briefly, how is an organism made? We have genes. The genes are the blueprint for the proteins. The proteins is what really makes our cells. So we have several stages. In reality, many more stages, but a simplified picture is this: DNA, RNA, it's a version of DNA that shuttles out of the nucleus and makes a protein outside the nucleus. So this is biology 101. And then there's an important word called gene expression, and the gene expression is something like how active is a gene. And that is not built in. That depends on your lifestyle, on all sorts of factors, random events, pollution, whatever, lots of factors. And there's a lot of research on gene expression because people are trying to understand which factors make people ill and how can we cure a disease by silencing genes and so on. So it's opened up a whole area in biomedical sciences. But now, thinking back about this testing, so this promise of selling you a test and telling you you will get, all these horrible diseases in 40 years down the road, has a few problems. One is the gene expression, because maybe the genes that they're finding there will never get active in your particular case, or you were run over by a bus before that even happened. So the question is, why is that information relevant if it is so far away? So here's basically my picture taken from the cancer world, now applied to this genomic testing stuff that they offer you. So you can live in a world where you don't do any testing, you have no treatment for these horrible diseases anyway, like Huntington's disease, you can't do anything about it, Parkinson's, you can't do anything about it when you know you have that gene. So at some point in your life you get a clinical diagnosis and then you die. So that happens to most of us anyway. Now in a world with testing, you can get a diagnosis and then you spend 40 years or so being really depressed about this, and then you also die. Same thing. And here, in this world, this is a slightly better world, because in this case, there was a treatment. So in this case, you were just depressed until treatment could start, and then you spend a good life, and then you even gain some time, and you die a little bit later. Yeah, so this is a good application for testing. So my recommendation to you is only do tests when there is some kind of action point, then some kind of treatment, not just to have more information, because we grew up in a world where we thought information is good, information is power, the more the better. And I think that is a bit outdated. So besides the usual reservations about those kind of test companies, you know, who knows what their research is built on, what population, does it apply to you, what about the measurement error, what about multiple testing, because if you test for you know, if you test for 20 conditions at 5% significance, you're going to be, on average, you're going to have one of them. So that is just the 5%. 5 times 20 is 100. So false positive rate in multiple testing is built in, basically. So I'm very suspicious, but if people, I know not everybody agrees. And of course, these Bay Area companies, they're very, very excited about their, we had the founder of them in the Sheldonian in Oxford, and it was really science fiction. It was quite extreme. But I admit there are also reasons. I mean, there are people who were adopted and, you know, have zero information about their family. So there are cases where I think that is a good thing to do. I'm not doubting this. But I thought it is good to have this kind of picture in mind before making a decision to ask for information that you would otherwise maybe not get. Then I was going to also talk a little bit about a project in behavioral finance, but there's not that much time, so I have to make a quick decision what I tell you about that. So I think I will just skip some of the background now and go straight to the project. You just have to believe me a little that people can be irrational, so I'm not going to give you the whole introduction about the possibilities that stock market traders or private people who trade on the stock market may not always do rational decisions. So if you can accept that, we can skip the background. There are lots of biases. If you go online, financial behavioral finance, they have a lot of biases. Here's a whole list of them. So it's fun to look at that, how irrational people can behave. Now, where is this? Yeah, so what I wanted to so that was the background. And because of that, there's a whole area that's called behavioral finance that looks at human behavior making financial decisions. And where's the start of that? Oh, okay, yeah. And so what I wanted to look at here is this project that I have with a student, and now it's another student on the project, where we look at selling behavior. So what you have, one of these biases that you have, is the so-called disposition effect. The disposition effect is that people hold onto their losing stock for too long. So instead, they tend to, this is just on average. Of course, if you know it's losing, you're not doing this, and so on. So it's irrational. So what happens is, it's a bit like the gambler's fallacy. People's stock loses, but they don't want to believe it's losing. So they're trying to rationalize that, and they're trying to justify, well, actually, I bought it for this money, that's what it's really worth, and it's not really losing, or they just try to forget about it. So it's just what has been observed. It's not what people think they're doing, but this has been observed that people just behave like that. So the way you check this is you look at a data set and you just compare how much is being sold at which level and so on, and how much is so how much is being sold as a gain and how much is being sold as a loss, and compare those times, and then you just find that empirically. And then you're stuck with the situation and you try to explain it. So we're doing a bit more research about the mechanism, why people do this, when exactly they do that, what does the trajectory look like for people who do that, and so on. So we're looking at the selling behavior. And there's already a bunch of other studies, and we did similar things. We looked at round trips, and we checked, for example, we looked at threshold strategies. So we look at, one version of people would follow a threshold strategy. That means they just sell things when they are above or below a certain value, and that would be a quite rational way of approaching that, provided your thresholds are good. But this is not what people are doing, because we have this, this plot here that actually shows that's not what they're doing. Because if they did that, then they wouldn't have all this time that they spend below the price that they end up selling it. So we get here a more complicated histogram, which is evidence against a threshold theory. So you compare it what price they end up selling it, and you find that they already spend time below that price beforehand. So that means they don't have a threshold theory strategy. So we did a lot of these plots, and it's fine. But suddenly, I realized that this kind of approach is getting one thing wrong. We are looking at trades because all these distributions we calculated were just based on round trips. So every round trip has the same weight in this histogram. But this is not what we are interested in. We are interested in people. And that's what I said. I realized a couple of months ago, oh my God, we're sampling on the wrong level. We need to sample on the level of people. I mean, it's a bit more complicated than the data set, because they're not, we need to match that all and so on. But first I mentioned that to someone, they said, oh, everybody does it that way. I think, yeah, that's not a good excuse. So we're going to get that right now. We're doing it again at the moment. But in the meantime, I just thought I should look into how much it actually matters, do some simulations, just for fun, to convince people that we should care about this. I mean, for statisticians, it's actually pretty clear. Once you realize what you're doing, it's pretty obvious. You have to, because we want to describe people's behavior, not stock behavior, so we need to sample people. It's only a problem if the people, so you have people who trade faster and you have people who trade slower. And if they were all roughly trading at the same speed, it wouldn't matter, because everybody would be equally represented in the pool of the trades. The problem only occurs if some people are oversampled because they trade so fast compared to others, and then it only matters if the outcome variable you are interested in actually has something to do with that speed. So it's not necessarily a problem, but it can be a problem. So that's why I think we need to just do that properly. And then I did here, just for demonstration of that, a few simulations where I created a fast trading group that used the threshold, the small threshold, small window, and a slow trading group. I made it a bit cheap here. I just made them deterministic and holding stock really long. And then I mixed them up, and so to compare the effect of that, so this is just the stopping time basically as a check if the simulations are working. So in this one, I just did the trade perspective. That means every trade counted the same. And here I calculated the average first for each trader within all the trades that trader did, and then I compared on that level. So that is the trader level, trader perspective. And here's a more interesting plot where I did the same kind of experiment with these groups, but now what I measured was the relative number of days below the lower bound, below the threshold. That's why it looks very boring for the threshold group, because they actually go out of the game. And here is the other group that stops at that deterministic time, so that is the proper distribution. If you mix them up, first you're a bit puzzled because what happened to this distribution? Well, then you realize the scale the scale changed. So that distribution is hidden here, very close to the x-axis. You can't see it. So that happens sometimes. It's actually not a programming error, it's just scaling. So if you mix them together again and average at the right level, you get this kind of mixed distribution from these two. So that's an example where you just need to be aware on what level you are sampling. So that's a bit different from the selection bias. It's like a hierarchical situation that needs to be recognized. Couple of comments about length bias, and selection bias as well. They can come in to quality assessment of items. For example, if you have some items that are that you can that deteriorate slowly, and others that deteriorate fast, it's like a cancer that grows fast or slow. So you can have that sort of phenomenon in completely different applications from cancer. I think we are going to apply it in the context of deterioration of X-ray detectors, where we have a project on damage on X-ray detectors, and that may vary a lot depending on whether they're used for medical purposes or whether they use them in the Diamond Light Source Center and put high energy on it, so they deteriorate much faster. And another important thing about sampling selection bias in a way can come in with the current trend to do a lot of crowdsourcing of data. Because if you do crowdsourcing of data without any quality standards, what can happen to you is that the repository gets swamped with very dirty data, because that gets in faster. So that's something to watch out for, and that's why usually after a while. And then that kind of gives the whole data repository a bad name, even though there may be high quality data in it. This just automatically happens, that cheaper data is faster, can be generated more faster, so that gets automatically overruns the repository if you don't have mechanisms to control for that. All right, so I hope that's been interesting. Come with questions if you have any, or suggestions.
Thank you. Extremely interesting talk. Just a quick one on the fastness. Now, for traders, I don't think you included transaction cost. So some fast traders, they churn 1,000% of the stock during the year, and it costs, if it's your pension fund, it's extremely expensive. So the fast traders are costing the country a lot of money.
Yes.
In an ethical point of view, and the same. So that maybe you should include cost, and maybe you should include cost also for the. I'm slightly nervous about you saying, well, don't get treated, because it could be in a period of austerity. Sorry to be politically, screening costs a lot of money. So that anything which says we don't, arguments for not screening, for screening slower, also has a cost implication.
Oh, absolutely. It's very sensitive.
Suggesting you put cost in some of your analysis.
Yeah, this wasn't so quantitative. This was just about the sampling issue, the fast and the slow. It was not actually saying one of them is better, faster, or slower. It was just trying to make the point that we observe something different. The screening is very, very difficult question.
You also said, sorry, dirty data gets in faster. So again, somehow the speed.
Yeah.
Speed can be good and bad.
Yes.
Depending on what the.
And also the screening, so there were things I only learned while we're doing this project. So in America, they screen age 35 for breast cancer. They start screening and tell people they need a baseline, things like that. But this is probably extreme. On the other hand, they only have one radiologist looking at the picture. In the UK, they start at 50 and only screen every 3 years. But they have two radiologists independently looking at it. So we have to be so careful when we compare different countries, because the settings can be very, very different. And I'm not really, actually, I haven't committed to any opinion myself. But I think you're also right that we need to be very careful with how things can be interpreted. As soon as you say something as a statistician, they can always pick the interpretation that's convenient for some objective, right?
I'll ask one question about thinking about the relation to some of the things in the workshop. Is that there's a lot of adaptive refinement in model fitting that goes on when we make model approximations, which is perhaps ripe for introducing a correlation between accuracy and the criteria you're interested in. So possibly introducing bias. So I would just suggest that perhaps if you're thinking about those sorts of fitting issues, you might have a conversation about sampling bias and what that might introduce into your final final model response criteria in the problem domain of interest. There's no other question. Oh, Michael?
Just one. So is this the reason why election posters usually get it wrong? Is that they have sampling biases. The people that they can sample are not, basically show all of the biases you've just shown.
I think so. I mean, I'm at Warwick, so we have this person, my colleague David Forrest, who does the exit polls, and he says, "I don't touch polls before the election." He wants to have nothing to do with it. I think, he probably has lots of reasons. For me, one reason would be there's a huge gap between what people intend to do or tell you they will do and what they actually do. Not just because of the time. I mean, they ask people a few days beforehand. Just in general, the saying I will quit smoking, saying I'll do more exercise, and actually doing it. I mean, that's a whole field in psychology is to study this problem, that there's such a huge gap between these two things. So for me, that's the biggest issue about polling. But there are lots of other issues as well. Usually they only get it right if they have a very similar historical situation, so they can just do a little bit of, it's still in the sensitivity of the old of the old result. There's so many famous examples where it went wrong when something historically was really different. On the other hand, we have learned a lot from this. If you think about the presidential Gallup, you're American, yeah, so you probably probably know that. Yeah, I mean the whole idea of selection bias goes back, I think, to Gallup realizing that doing a telephone survey in 1936 was really a bad idea. So they had a sample that was only a fraction of that telephone sample. They did postcards, but they had a representative population. Yeah, but I wouldn't choose to do election forecasting.
Let's thank the speaker again.
Thank you.